Howdy, this is Mr. Justice. In this video, we're going to talk about the draw notes that we created in class on day 34. So take a look here. This is the overview of what it's going to look like once you finish yours. I've also put a copy of this PDF, a PDF copy of this on the notes page as well, so you'll be able to access that if you want. And my expectation is that you're following along and taking notes as I go through this. So allow me to zoom in a little bit. So let's go through this handout. At the very top, we're talking about comparisons, the differences between anaerobic and aerobic respiration. The term anaerobic refers to an environment that is without oxygen gas, whereas an aerobic environment includes one that has oxygen gas. All right. So what I'd like you guys to do is take your paper. We are taking our paper and we're going to make our notes horizontal like this. All right. So make sure that you can fit everything on the page. And what I'd like you to do is about 2/3 over from the left of your page, we'll make a nice dashed line that you're seeing on the screen. What we'll do is we'll label the side that's a little bit larger, right? The side's a little bit smaller. There's that dashed line. So how about we put the anaerobic respiration right at the top, and we'll mention where this occurs. This process occurs in the cytoplasm of the cell, so outside of the mitochondria. So anaerobic respiration is the very first step here in the overall total oxidation of glucose. And this does not require oxygen, this process here that we're about to talk about. On the other side of that orange dashed line that you're seeing on my screen, I'd like you guys to write on your paper aerobic respiration. And let's write down the location of where this happens. This process here that we've been talking about, and that includes the Krebs cycle, electron transport chain, oxidative phosphorylation via ATP synthase. All of this process happens in the mitochondria. All right, so we're going to go back over to this side here. So what I'd like us to do is write out 6 carbons. How about we link all of these 6 carbons together in one long chain? And what that will do is represent glucose, which has a chemical formula of C6H12O6. And so each of these carbons then represents one of those 6 carbons in glucose. And then what I'd like us to do is draw a line from that 6 carbon chain. And at the end of that is well, take a look, 2 3 carbon chains. There's 1, 2, 3. And then here's the second 3 carbon chain, 1, 2, 3. This process of breaking or splitting apart glucose is called glycolysis. This whole process is aided by, catalyzed by, facilitated by enzymes found within the cytoplasm. This process takes a little bit of an investment in energy to get it going in order for these enzymes to be able to split glucose into 2 3 carbon products. That energy comes from the 2 ATP that power this process. That initial investment of 2 ATP molecules is going to yield 4 ATP molecules and 2 NADH energy molecules holding those high energy electrons. Students, because we used 2 ATP to make 4 ATP, we net 2 ATP. And we'll put that into our notes. So what are the products then after glycolysis? After we split the 6 carbon sugar, we'll have what are called 2 pyruvic acids, as represented by these 2 3 carbon chains that you're seeing on the screen. Now, something can happen here. I'm going to cover this up so that we can, let's go ahead and do that. There we go. So we can kind of direct our attention here. So now listen, if oxygen's present, if O2 is present and the cell has a mitochondria, these pyruvates are going to be further oxidized and proceed into the mitochondria, where they'll be modified into acetyl-CoA, and then those organic compounds, if you recall, are going to be stripped of their electrons through the Krebs cycle. Those electrons are stripped off, they move down the electron transport chain, and poof, we make ATP. How much ATP? We're going to produce about 34 to 36 ATP during aerobic respiration. This requires oxygen to happen. And as we totally oxidize and break down glucose, glucose into, well, what are the products? Into what? Carbon dioxide and water. Now, this again requires oxygen to make sure that these steps move forward. The steps involved with aerobic respiration, when oxygen is present, pyruvate will be modified and stripped of its electrons during the Krebs cycle. Those electrons stripped from pyruvate and acetyl-CoA and the different organic compounds associated with the Krebs cycle, those electrons will move down the electron transport chain. Those proteins embedded within the inner membrane of the mitochondria will pump H plus protons into the inner membrane space, and then those H plus ions will zoom through ATP synthase that is able to capture the kinetic energy of those moving H plus ions and harness that energy to produce ATP. How much? 34 to 36. The bulk of our ATP production is happening when oxygen's present in the mitochondria. Now listen, what we're talking about here today though is the differences between aerobic respiration and anaerobic respiration. So sometimes oxygen is either not present or not required by the organism to oxidize these pyruvates to harness the energy. So if no oxygen is present, the pyruvate will enter into one of two processes, and those processes depend on the organism that does it. One of the processes is called alcohol fermentation. The NADH produced by glycolysis is used to help enzymes modify the pyruvate into ethanol. This ethanol is represented by these two carbons right here. In addition, we're also producing carbon dioxide. So the organism that is able to undergo alcohol fermentation is yeast. And brewers and bakers use yeast's ability to undergo alcohol fermentation for their own processes. This ethanol is what makes it into wine and beer and spirits. And if you're a baker, this process is going to produce a little bit of ethanol in your raw dough, and the carbon dioxide produced during this process will help make that dough rise, creating all of those bubbles that you see inside of bread. And then when you bake the bread, the ethanol just evaporates away, so it doesn't even a part of it. But there you kind of have that essence of something special that you're eating because of that effervescence of the ethanol. So yeast undergo alcohol fermentation. Some other organisms, including us, will undergo lactic acid fermentation. We still need enzymes. We need those two NADH produced during glycolysis earlier. And what we're going to do is transform that pyruvate into a 3-carbon lactic acid. Now, this process occurs by bacteria like lactobacillus. These bacteria are used by food producers to create yogurts. But we also do this too. In fact, our muscle cells undergo lactic acid fermentation. And you actually feel that if you ever have exercised before and really pushed yourself so that you are really breathing really hard. What's going on is that your body and your muscle cells are not getting all of the oxygen they need to fully oxidize glucose and make that 34 to 36 ATP. But those muscle cells are still working. They still need to get ATP. So for us, our muscle cells will undergo lactic acid fermentation. We might make only about 2 ATP while we're doing it, but we're also producing lactic acid as a waste product. And that lactic acid you can actually feel in your muscles. Sometimes you'll feel it in your diaphragm, which is a muscle underneath your lungs. You might feel it in your legs if you're really pushing yourself. But yeah, this process is also done by bacteria, and that's something interesting, right? This process of glycolysis is something that we have in common, humans have in common with every living organism on Earth, all the way down to bacteria. It's a process that has been around for billions of years. So let's take a look here. We've got 2 types of fermentation. Alcohol fermentation is performed by yeast. The NADH made during glycolysis is used to transform pyruvate to ethanol and carbon dioxide. Bakers and brewers use this process to their advantage. Lactic acid fermentation is performed by bacteria and animal cells, including the muscles in humans. The NADH made during glycolysis is used to transform pyruvate into lactic acid. Pretty wild stuff. That's the finished product, guys. Make sure that yours looks just like this, and I hope it was helpful.